PCR is a common lab technique used to make copies of a particular region of DNA. The goal is to make enough DNA that can be analysed or used in some way. Researchers might want to study the function of a gene. Forensic scientists might want to match the crime scene DNA with a suspect. So, what is required for PCR? Well, you need the DNA that is to be copied. This could possibly be from a sample of hair left on the body of a murder victim. You will need a thermocycler. This is a massive piece of machinery that you will stick the vial containing the solution into, and it can raise and lower the temperature of the sample. PCR requires a polymerase enzyme to make new strands of DNA, just like in normal DNA replication. The problem is, in PCR, heat is used instead of helicase to split the DNA up into two strands. At high temperatures, most enzymes do not work very well, or at all. The polymerase typically used in PCR is Taq polymerase, named after the bacterium in which it was found, Thermus aquaticus. These bacteria live in hot springs and hydrothermal vents, which means they have to be tolerant of high temperatures. Their polymerase is most active at around 70 degrees centigrade. Taq polymerase can only make DNA if it is given a primer. A primer is a short sequence of nucleotides that provide a starting point for DNA synthesis. The primers are single strands at about 20 nucleotides in length. They are given a sequence that will make them bind to the opposite strands of the DNA template of the edges of the region that you want to be copied. When they bind, they will be extended by the polymerase and the region between them will get copied. Nucleotides are the DNA building blocks that are what copies of the DNA will be made out of. There are 3 steps to PCR. The DNA is heated to separate or denature the DNA strands, which means they're single-stranded. The reaction is cooled so primers can bind to their complementary sequences on the single-stranded template DNA. Raising the temperature again, the Taq polymerase extends the primers and synthesizes new strands of DNA. The exponential growth of PCR. The cycle repeats 25 to 30 times in a typical PCR reaction. This can take around 2 to 4 hours depending on the length of the DNA. But the DNA is copied exponentially. In each cycle, it's not just the original DNA used as a template, but the DNA made in the round before. So you have 1 piece of DNA, then 2, then 4, then 8, etc. This is why with PCR you can get millions or billions of copies of the DNA sample. This is enough copies of the DNA to be analysed using other techniques, for example, visualised by gel electrophoresis.